Hello, good morning. Myself, I am BSR, botany lecturer. Today, our topic secondary growth. Secondary growth generally seen in dicots and gymnosperms, not seen in monocots. But in some monocots like Dracaena and Yucca, anomalous secondary growth is present. So, before going to secondary growth, the vertical growth of the stem, that may be apical meristem present at the stem tip and root tip. This is apical meristem. Vertical growth of the stem carried out by apical meristem. Whereas in the dicot plants, especially in the dicot stem, we are discussing the secondary growth. So they're increasing in the thickness of the stem. Increasing in the thickness of the stem or radial growth of the stem is called secondary growth. Generally, secondary growth carried out by lateral meristems. Anyway, this secondary growth we can discuss by two steps: intrastellar secondary growth and extrastellar secondary growth. Intrastellar secondary growth. So the secondary growth inside the stele is called intrastellar secondary growth. So when you observe in the intrastellar secondary growth, look at here, this is the dicot stem. Yes, this is the dicot stem. Just in the dicostem, the vascular bundles are arranged in the form of ring. Vascular bundles are arranged in the form of ring. That condition is called eustele. Here the vascular bundles are conjoint, collateral, open vascular bundles. Conjoint means xylem, phloem constitute to form one bundle called conjoint. Present on the same radius called collateral. In between xylem and phloem, cambium is present. Cambium is a meristematic tissue. Due to the presence of cambium, that vascular bundle is open. So they are conjoint, collateral, open vascular bundles. Conjoint, collateral, open vascular bundles. Especially open means cambium. Cambium means meristematic tissue. So, especially the meristematic tissue present within the vascular bundle is called intrafascicular cambium. Intrafascicular cambium. Intra, within the vascular bundle. This intrafascicular cambium is a lateral meristem, but it is not secondary meristem. Intrafascicular cambium is a lateral meristem, it is not secondary meristem. Remember that one. Whereas when you observe in the middle stem, their medulla is present made up of parenchymatous cells. These parenchymatous cells are permanent cells, that one. Whereas these permanent cells, parenchymatous cells are present between the vascular bundles is called medullary rays. It's a medulla. This one is a medullary rays. It is a permanent tissue. Sometimes the permanent cells undergoing dedifferentiation. It is undergoing dedifferentiation to form meristematic tissue. To form meristematic tissue. So this is called interfascicular cambium. It is interfascicular cambium. Interfascicular cambium is a lateral meristem and a secondary meristem. It is lateral meristem, it is not secondary meristem, but it is lateral meristem, it is a secondary meristem which forms later. It is not present in the embryonic stage. So that is interfascicular cambium. So intrafascicular cambium, interfascicular cambium fuse to form a vascular cambial ring. This is the vascular cambial ring. Now vascular cambial ring is formed. What is the activity of vascular cambial ring? Which forms new cells towards inner side called secondary xylem, and which forms new cells towards outside, it is called secondary phloem. So to understand very easily, look at here. So this is the vascular cambium. This is vascular bundle. It is the vascular cambium. So here secondary xylem. It is the secondary xylem, and here secondary xylem, it is a primary xylem. So when the secondary xylem forms, that is secondary xylem pushing the primary xylem towards the center, like this, towards center. So when you take, this is the secondary phloem. So when the secondary phloem forms, which pushes primary phloem towards the outside. So always secondary phloem and secondary xylem present on either side of the cambium. This is the cambium. So it is the intrastellar secondary growth. So the vascular cambium always forming secondary xylem and secondary phloem. Now you should know what is the mean of spring wood and autumn wood. Very simple terms. Spring wood means early wood. Autumn wood means late wood. Autumn, spring, early. In the spring season, you know, in plants, more number of leaves and flowers are present in the spring season. When more number of leaves and flowers are there, they require more amount of water. When more amount of water is required, more number of xylem vessels with wider luminis forms. So look at here. This is the vascular bundle, and you take, this is the cambium. Here, spring wood, it is the primary xylem. In the spring season, so more number of xylem vessels are forming with wider lumen. So xylem vessels with wider lumen. So this wood is called spring wood. This wood is called spring wood. Whereas in the autumn season, late wood, in the autumn season, less number of leaves are present, less number of flowers are present. So it forms less number of xylem vessels with narrow lumen. So this wood is called autumn wood or late wood. It is autumn wood. So the 1 year is over. It is a spring wood and autumn wood. Again in the next year, there will be a spring wood. It means more number of xylem vessels with wider lumen. Spring wood. And autumn wood is autumn wood. So every year there will be a spring and autumn wood. These rays are called vascular rays, or these rings are called annular rings. So by counting these annular rings, so spring wood, autumn wood, again in the second year spring wood and autumn wood, by counting these annular rings, the age of the plant can be estimated. That science is called dendrochronology. So this is spring wood and autumn wood. So in the autumn wood, the less number of xylem vessels with narrow lumen is present. In the spring wood, more number of xylem vessels with wider lumen is present. You know, this is the secondary xylem. The secondary xylem consists of vessel elements, xylem fibers, xylem parenchyma is present. Like that, when you go to in the secondary phloem, sieve tubes and companion cells, phloem fibers, phloem parenchyma is present. You know that one. So it is a spring wood and autumn wood. Then you go for heartwood and sapwood. What is the mean of heartwood and sapwood? So you take, this is the stem. I am observing at the center of the stem. Here heartwood. Heartwood is the center of the stem. So it is the earlier formed secondary xylem. Earlier formed. It is present at the center. Earlier formed secondary xylem is called heartwood. This heartwood is durable, more durable, which containing ergostic substances, tannins, resins, gums are present, which is not at all useful for conduction of water because it is blocked by tyloses. Xylem parenchyma forms tyloses. It is not useful for conduction of water. Totally tyloses are present like this. So earlier formed secondary xylem is called heartwood. It is not useful for conduction of water. Whereas recently formed secondary xylem, it is recently formed. This wood is called sapwood. It is a sapwood. Sapwood light in color. It is light in color, whereas it is dark in color due to the presence of ergastric substance. Sapwood helpful for conduction of water. It is helpful for conduction of water. It is not helpful for conduction of water. Sapwood containing both spring wood and autumn wood. Heartwood containing both spring wood and autumn wood. So you know what is the difference between hardwood and sapwood. Then we are going to extrastela secondary growth. The secondary growth outside the steel. The secondary growth outside the steel is called extrastela secondary growth. You can understand very easily. So look at here, this is the vascular bundle, and you take this is the cambium, this is primary xylem totally crushed, this is the secondary xylem. So when more amount of secondary xylem and the secondary phloem, here this is the secondary xylem, and you take this is the secondary phloem. When more amount of secondary xylem and secondary phloem forms, the vascular bundle size is increased. This is the vascular bundle. To understand only I am telling here in competitive mode. So vascular bundle size is increased. When the vascular bundle size is increased, the primary protective layer called epidermis, that epidermis is ruptured because the vascular bundle size is increased. Primary protective layer is ruptured. So that is why a secondary protective layer is formed. So formation of secondary protective layer is called extrastela secondary growth. So below the epidermis, their cortex is present. The cortex also made up of parenchymatous cells. They are permanent cells. Those cells undergoing dedifferentiation to form a meristematic tissue. That meristematic tissue is called cork cambium. This is cork cambium. Cork cambium is a lateral meristem. It is a secondary meristem. Now the cork cambium forms the cells towards outside. Some of the cells towards outside. Some of the cells towards inner side. So the cells forming towards outside and some of the cells towards the inner side. So the cells which are formed towards outside is called cork. The cells which are formed towards inner side is called secondary cortex. Cork is also called phellum. Cork cambium is also called phellogen. Secondary cortex is also called phelloderm. Phellum, phellogen, phelloderm constitute called periderm. So formation of secondary protective layer. What is the secondary protective layer? Periderm. That is called extrastela secondary growth. And important here, remember what is the mean of bark. Outside the cambium, whatever tissue is present, outside the cambium, that is secondary phloem to cork, is called bark. Up to this. Outside the vascular cambium, whatever tissue is present, that is called as bark. And one more important point, these cork cells are dead. So in the cork cells, there some thin lens-shaped cells are formed. That thin lens-shaped cells are called lenticels. These lenticels helpful for exchange of gases. So this is extrastellar secondary growth and total extra and intrastellar secondary growth we discussed. Thank you to all.